Okay, so welcome to the neuroscience, neurophysiology portion of Pathophys. So you'll have to, having got used to Dr. Lindblad, you'll now have to sort of shift gears and get used to me instead for the next 20-odd classes. So we'll be spending the first couple of lectures, about 5 lectures, doing kind of an overview of like a kind of superficial summary overview of neuroscience before we kind of delve into sort of more neuro-related disease states. But that's just so you kind of have some background in order to understand what's going on in some of these disease states. And just as a sort of follow-up really to the exam you just took, which I gather you guys overall did really well in. So you may know that sort of many of us are sort of in the middle of this sort of whole year-long teaching course. And so one of the things that sort of is kind of stressed is, different aspects of, basically how to help you guys sort of, study better. Because obviously as far as success goes, one half of the equation is obviously me doing the best I can in terms of explaining things so that you kind of get it and find things interesting and so on. The other half of the equation is you guys finding really effective ways of learning stuff. And so on the Canvas site, Dr. Phipps has posted this video from Dr. Sandra Maguire, which is, I was not going to play for you because it's a full class. It's about 45 minutes, but she makes a lot of really good points about how to study effectively for essentially a difficult subject. She's a chemistry professor, and her way of looking at things is that if you do everything she says, it is still possible even in chemistry to achieve 100%. And so many of the same ideas are going to be true for sort of things like pathophysiology and other subjects you're going to tackle in pharmacy school. And one of the key points, or the sort of two of the key points basically, is if you think about studying to teach versus studying to learn. Like if you have like a big test tomorrow and you're going to be trying to figure out how you're going to go about tackling it, right? You're going to look at your notes and you're going to read through the notes lots and lots of times. However, if you weren't taking the test tomorrow, but your best friend is taking the test tomorrow, you'd approach the studying a little bit differently, right? You'd be sort of taking that little bit slight step back and try to figure out, almost more sort of dispassionately, what's likely to be on the test? How can I explain that better? And so if you approach your studying sort of in that way, like you're basically trying to help your best friend study for the test rather than yourself, you might find that it's actually a more effective way of, getting that sort of information kind of lodged into your head. And the most important thing is, or one of the other really important things is just always believe that you can improve, because, there's never any kind of ceiling on what you can sort of achieve in all of these courses. Especially when you're starting pharmacy school, there's this big, kind of cliff of, it's a significant step up in difficulty to what you guys have been used to. But that doesn't mean that it's sort of going to be an impossible task. It's just a matter of kind of figuring out how to deal with it and tackle that kind of learning problem. And this is just a sort of, one of the sort of suggestions for sort of how you might structure learning time, studying times. And so, some level of quick previewing the material for a class, can help, because it can just get you I'm, at least as far as I'm concerned, I'm not saying sort of read the chapters and things like that, but just have a sort of vague idea of what will be talked about so that you're just receptive right when the class starts. And a big thing is obviously attending class, because that gives you something to hook your memories onto, and then do some level of review as soon as possible after class. And one of the sort of new things I'm doing this year is that at the end of each class, at the end, and you'll see it with this current slide set, there's a sort of list of, I think it's like, I don't know, 10, 11, 12 questions, something like that. And so those questions, if you can really effectively answer all of those questions, then you'll have got a lot out of this class. And so, it's not, I won't say that every single exam question I set will be one of those questions, but the majority of them will be. So, yeah, like I'm not saying, don't look at the lecture notes. The whole point of the questions is to try and give you like a lens to look at the lecture notes, so that if you sort of are viewing it that way, then you can help, that will hopefully help you sort of be prepared and get as much as possible out of all of these classes. So it's the idea is to sort of, like I'm trying to sort of help you guys help yourselves with trying to get as much as possible out of all of these classes. So that being said, we can move on to the sort of main subject. And I guess I should also apologize for wearing this daft headset, but this is the best way of getting some sound recorded on the Zoom videos, because otherwise I'm stuck literally right in front of the laptop. So at least this way I can pace about like 3 feet either side of it. So exactly. So anyway, so I will pretty much every class I should have a poll everywhere question, and that's the easiest way for us to keep track of attendance to classes. And so I really encourage you to actually sign in and respond to these poll everywhere questions. And in this case, it's a straight, sometimes I'll have like little questions about, say, the previous class or the previous section we've just talked about. In this case, it's just a simple question. So I'm going to teach you about neuroscience. What does that have to do with pharmacy school? So if you could quickly sign into poll everywhere, just so because of this. What's that? Oh, no, it's a... Oh, I see. That is funny. And I think this would be a great poster, sort of, because I think we have pretty much most of the things that I would think of. So drugs is the big one, and that makes sense for pharmacy school. If you sort of look at the sort of list of most highly prescribed drugs, obviously you'll see things like blood pressure medications and so on. But things like antidepressants and things like that are also extremely highly prescribed. And so understanding how different drugs interact with the nervous system is obviously crucial to sort of modern medicine. And in fact, it's more than just sort of, like what drugs you're giving in order to treat certain neuro-related disorders. You also have situations where certain types of drugs, might not be intended to hit the nervous system, but they still do. And so you're also very much concerned with side effects and things that interact with, for example, the cholinergic nervous system are, like a major area of drug side effects. So this is pathophysiology, not neuropharmacology, which we'll be tackling next year. And so we're more focused on the actual disorders rather than like the treatments. And in some cases, I'll be talking about some neurological disorders that don't really have any kind of treatments currently. But this is just a chart showing different types of neurological disorders. And what I've done is sized the surface area of each of those circles to give the relative frequency of those in the US population. And what you can see is migraine is the most common neurological disorder. And so that's because it does affect sort of, like a very high proportion of the population. And in many cases can be fairly disabling. Other big ones are going to be stroke, Alzheimer's disease, Parkinson's disease, epilepsy. And this may slightly surprise you, even schizophrenia is actually remarkably common in the overall population. In many cases, it can be very effectively controlled through medication, so that, people live, like entirely normal functional lives with no problems, as long as they sort of remain, well controlled for their symptoms. There are going to be other disorders that we also talk about, for example, multiple sclerosis, myasthenia gravis, ALS, Lou Gehrig's disease, and so on. They're less common. In many cases, some of these things we actually talk about because they actually provide additional insight into sort of how the nervous system actually works. For example, myasthenia gravis is a disease of the neuromuscular junction, and by understanding the disease state, it really gives you a good insight into sort of how neuromuscular transmission works. So we're starting right at the top with some basic sort of definitions of, about what are the parts of the nervous system. Can't get more basic than that. And so we have our neurons. They connect to each other with axons, and the actual site of that connection is going to be a synapse. Then we also have glial cells, and then we have the structural components that act to support and protect the other portions of the nervous system. So we have the meninges, which are these layers that sort of are basically surrounding the brain. And we also have all of the blood vessels that penetrate and feed the brain. And these are sort of also play an important part in something else that we'll talk about, which is the blood-brain barrier, which is important for separating the nervous system from the rest of the body. And all of these different components of the nervous system are obviously, vulnerable to different types of insult. So obviously there's straightforward disease. There can be sort of genetic factors which can affect the nervous system, whether they're sort of things that are directly affecting different biochemical pathways within the neurons, or affecting the migration of sort of newborn neurons during development, so that there are changes in overall brain structure. There can be changes, sort of mutations in the some of the signaling proteins within the neurons, which result in different types of disorders, especially things like epilepsies. You can have environmental insults that affect especially the developing brain. And then you can have just direct trauma causing problems. So for example, like an obvious one is going to be a head injury is going to cause serious problems, but also things like strokes, like that's some kind of insult to the blood vessels, whether it's bursting them or blocking them, and that will lead to damage to the nervous tissue that's supplied by those blood vessels. And sort of over the course of the next few weeks, we'll sort of be talking about different types of neurological disorders which have different aspects of these different types of insult. So, starting at the beginning, the basic unit of the nervous system is going to be the neuron. And so here's a cartoon. Here is an actual neuron. This is a hippocampal neuron. And the basic architecture of a neuron is that there is going to be, at some point, a large swelling, which is the cell body. And that's the site of the nucleus. And it's also where the majority of the protein synthesis and things like that occur. Then you have the long projection from the neuron, which is the axon. That's the sort of output of the neuron. In this three-dimensional reconstruction of the neuron, you can see this one smooth process here. That's the axon of this neuron. So that's the output of the neuron. And so that will then project out, and then it will either contact another neuron and talk to that neuron, or it will contact some kind of target. So that target could be muscle fiber, in which case it's like going to be a motor neuron and sort of telling the muscle fiber to contract. Or it could be contacting say an organ like the heart or something like that, and then telling the heart whether to beat faster or beat slower, things like that. So you have the center of the neuron, the cell body. You have the axon for the outflow. Obviously, you need some way of sending information to the neuron, because otherwise the neuron would just have to kind of make up everything as it went along, and sort of that probably wouldn't be the best way of running a brain. And so you need inputs, and the inputs are these in the 3D reconstruction. These are these knobbly projections that are coming off the cell body, and these are the dendrites. So these are the sites at which other neurons are talking to your first neuron. And the dendrites are the sites of the synaptic connections. So we have the axon from one neuron contacting the dendrite of a different neuron to make a synapse. And the synapse is where the neurochemical transmission occurs. And that we'll be talking a little about. I think it's Wednesday, I guess it is, because there's no class Monday, Tuesday. So we have this very sort of specialized architecture, but and neurons come in all kinds of different shapes and sizes, but they all have these general features of an axon, a cell body, and a dendrite, and synaptic connections between them. So this is summarizing much of what I've already said. So we have our dendrites, where they're receptive regions of the neuron. We have the axons, which are the outputs of the neuron. And they come from a portion of the neuron, which is called the axon hillock, which is actually where the action potential is generated. And the action potential we'll be talking about in Friday's class. But the action potentials are the electrical signals that you can, in many cases, sort of record from neurons. And they're also what give rise to the electrical signals you see in, for example, an EEG. And they arise at this axon hillock of the neuron, and then they only propagate in one direction, which is away from the cell body. So we have this directional information flow. You receive information to the dendrites and send it out through the axons. It never goes the other way around. And as I mentioned, there are many, many different types of neuron, whether you're talking sensory neurons, whether you're talking inhibitory neurons, whether you're talking motor neurons. There's probably something like, depending on how you do your classifications, I'm sure you could easily get to 1,000 different types of neuron just within sort of humans, say. And then if you imagine how different neurons look in, say, a squid, then there really is a vast array of different types of neurons that one might study. But neurons alone are only one portion of the brain or the nervous system, and because the other portion that plays an important role are the glial cells. And so in the central nervous system, we have astrocytes, microglia, ependymal cells, and oligodendrocytes. And ependymal cells we don't really need to worry about. They line the surface of the ventricles and help maintain the flow of fluid through the brain. But they don't have, at least currently, don't have very much clinical significance other than, they're there and they work and help keep things happy, but there are no diseases that specifically impact them. Glial cells, other types of glial cells are much more, play more specific roles. And so they do actually play a role in modulating the signaling of the neurons, but they also play a major role as essentially providing support for the nervous system. And in the human brain, they're roughly the same number of glial cells as there are neurons. Although that will, vary from brain region to brain region. So we'll start with the astrocytes. So they're called astrocytes because they're star-shaped. And they have, well, this is a picture of one. And they have all these processes that kind of branch out. They kind of look a little bit like a neuron because they have these processes. The difference is these sort of, just to quickly tell at a glance that it's not a neuron, is these processes are sort of much thicker and kind of more sort of like blobby. Like more like, like kind of imagine an amoeba sort of kind of spreading out its little processes. They look a little bit more like that. They're little bit less structured. And these processes radiate out through the neural tissue. And what they do is they're there to basically regulate the environment around the neurons. So they send processes out that sort of envelop many of the synapses so that they can play a role, for example, in mopping up excess neurotransmitters. They also, as we'll talk about in a moment, extend out to the blood vessels themselves, where they both play a role in the transport of nutrients from the blood to sort of deeper within the neural tissue, and play a role in actually establishing the blood-brain barrier. And overall, they have this kind of regulatory role where they're acting to sort of keep things stable for the neurons, so that because the neurons themselves have these sort of very fixed structures, then they need something to sort of basically help move nutrients from the blood to the neurons, and then remove things that are unwanted, and in order to excrete them out into the blood. The next major type of glia is the microglia, and they're the sort of oddball ones because they're actually more closely related to immune cells. And I know you've just had a couple of weeks of immunology with Dr. Lindblad, and so developmentally they have sort of a lineage which is related to that of typical immune cells. They're not related to the other glial cells, and but they sit within the brain. And of course, the brain is encapsulated by this blood-brain barrier that usually prevents immune cells from interacting with the brain. So the microglia are the sort of the brain's own immune cells. So they sit within the neural tissue. And most of the time they're sitting, they have these sort of, again, these long processes that go out to kind of monitor the environment around them. However, in their case, these processes are there for a monitoring role. So that if they detect some kind of insult, then it activates these microglia. They pull up their little processes, and then they migrate to the site of wherever something bad is happening, so that they can basically initiate an immune response within the brain, using all the kinds of cytokines and interleukins and whatever that you've already recently been talking about. And then they phagocytose sort of like diseased tissue and things like that. And then they also play a role in resolving that immune response. And then they kind of go back to their sort of little star-shaped life before. And there is increasing clinical interest in them these days because there's increasing evidence that they actually do play a major role in things like Alzheimer's disease, and also in the pathologies of things like stroke and traumatic brain injuries, because they're initiating and resolving these immune responses within the brain. And because the brain is such a delicate organ, anything that goes wrong with that can have significant consequences for neurological health. So now we come to the oligodendrocytes. And the oligodendrocytes have a particularly interesting role in terms of how they interact with the signaling of neurons. So what they do is wrap the axons of the neurons. And they're called oligodendrocytes within the central nervous system. In the peripheral nervous system, they're called Schwann cells. And there are some differences between them, but sort of functionally, they play pretty much the same role. So what they do is they wrap they sort of go around the axons of the neurons. And by doing that, they, and this is something we'll be talking about on Friday, they enable faster propagation of the signals along the axons. And because, and that can sort of be 10 to 100 times faster because of the actions of these oligodendrocytes. And so the more you can speed up, obviously, the signaling between neurons, the more obviously you can speed up your thinking and reacting and so on. And so this is why there has this very, very specialized cell type has developed. And the majority of the axons within the central nervous system are myelinated, because that myelination is this process of wrapping the axons. And interestingly, this is something that actually occurs in humans, actually takes a long time in development. So when in for example, teenagers, that's the point actually where some of the last portions of their brains are actually being fully myelinated. The axons are being fully myelinated. And so that those sort of new kind of the additional connections that are occurring within the brain of teenagers is sort of one of the hypotheses as to sort of why teenagers sometimes can be a little bit crazy, because they're suddenly, gaining new connections with the brain and sort of much faster connections to certain portions related to sort of impulse control and so on. The other aspect of oligodendrocytes that's really worth remembering for a future class is that this is where in multiple sclerosis, the majority of the damage occurs. It's that the essentially, sort of in a nutshell, what happens is that immune systems from outside break in across the blood-brain barrier, attack the myelin sheath of the axons, leading to problems with axon propagation between neurons, leading to obviously all of the symptoms of multiple sclerosis. And we'll have a class on that in a couple of weeks' time. So now what I'd like is for you to spend 2 minutes in sort of groups of 2, 3, something like that, talking about what you think are the main takeaways from this little section. And if it works, this will hopefully be a 2-minute timer. Yes, it is working. Okay, so what things have we come up with?
The gross anatomy and function of various types of central nervous system. Neurological cells as well as glial cells. The astrocyte acts as cleanup crew, kind of taking out excess neurotransmitters and maintaining the internal integrity and the homeostatic level of the central nervous system within the blood-brain barrier. A whole bunch of other stuff. I'm sure other people have things to say.
I think that was a good summary. Would anyone like to add a little bit to it? Okay, okay, sure. Pretty much the same thing. So I'm there. I can't think of any other kind of glial cell that's not a neuroglia. So, but sometimes so sometimes you get in the habit of using different terms, and so that's basically my view of the breakdown. So I should probably have just used one term. I hadn't actually noticed that it kind of broke down. So, yes. Yes, absolutely. Because without like a basic understanding of the fundamental workings of the nervous system, like how can we ever like hope to fix things that go wrong other than by sort of, like random luck and finding, like something that does work, which especially in neuropharmacology is actually how a lot of the drugs were developed. But in an ideal world, we'll have everything like really figured out before we do these studies. Yes. No. So they are nervous associated. So like I said, in the peripheral nervous system, we have Schwann cells, which, but yeah, like so if you're if you're looking away from sort of neuronal cells, yes, there are. You like the heart isn't full of glial cells or something like that. Yes. Yes. Like how many of them are there per axon? Is that your question? So that will depend on the number of axons that neuron has and how many branches it has and how long the axon has. So if it's a really short single axon, then yes, one oligodendrocyte could probably do it. But if it's more complicated, then you'd need more. But it's not like a one-to-one relationship. And you can also have situations where you have one oligodendrocyte that actually does wrap around axons from multiple neurons too. So, okay. So now we're going to sort of have a quick overview of neuroanatomy so that when I'm talking about, say, Parkinson's disease and, like substantia nigra and the autonomic nervous system and the hypothalamus and things like that, you at least have somewhere to sort of start understanding, like what I'm meaning by those things. And so this is the sort of overall organization of the brain and the spinal cord. So obviously someone's head and the brain in the middle. It's always a good sign if there's a brain there. And what this also shows is these layers that encapsulate the brain and the spinal cord. And this encapsulation of the brain and spinal cord is actually fully continuous. So the layers around the outside of the brain, which are the meninges, actually do extend all the way down the spinal cord. So it's all one continuous fluid compartment. This is why, for example, if you're concerned about, say, meningitis or multiple sclerosis or something like that, you can actually do lumbar puncture to analyze the fluid in the spinal cord, and that tells you something about what's going on actually within the brain itself, because it's all one continuous fluid compartment. I could imagine. I have not volunteered to have one, so. And so this is and then obviously we have the skull, which is on the outside of those the dura and meninges. So this is kind of zooming in on that layer that's outside of the nervous system. And what you'll see is there's sort of, it's not just a single sheet, it has multiple components. So here, this is obviously the gray matter, this is the brain itself. And then right above it, we have the subarachnoid space and the pia. And the pia is just that a little sheet of cells which are just on top of the gray matter. So the brain side of the subarachnoid space. Then we have the subarachnoid space itself. And that's where the CSF, the cerebrospinal fluid, is circulating. Then, as you'd guess from the subarachnoid space, the next layer is called the arachnoid membrane. And then above that, you have the dura. And so the dura, the arachnoid, and the pia are the three layers that are regarded as forming these meninges. And that's the sort of, for example, meningitis is obviously an inflammation of the meninges. And then obviously above that you have the skull and its bones, so I'll let Dr. Phipps talk about that. So he's would definitely be of the view that the sort of probably nervous system is the second most important part of the body after the bones. So, so we have the meninges, and then we have the subarachnoid space, and then we have the cerebrospinal fluid, which is obviously circulating around and like down through the spinal cord. And that is a sort of physical sort of encapsulation of the central nervous system. Don't confuse it with the blood-brain barrier, because it's not the same thing. The blood-brain barrier is the barrier that prevents things from going from the blood into the nervous system. And so the blood-brain barrier goes all the way throughout the brain, because obviously we have blood vessels that go all the way throughout the brain. Because otherwise the center of your brain would never get any oxygen, and you just have this very tiny little thin layer of workable brain, and everything else would be kind of wasted space. So we have blood vessels that penetrate through the brain, and if this was a medical class, for physicians, you would be probably have like a week just on the anatomy of the blood vessels that go all the way through the brain. So that, because they have, but you don't really need to know that, because I'm assuming none of you are going to become surgeons. So we're sort of, kind of leave out a lot of the anatomy, but you do need to know that there are these complicated branching patterns of blood vessels penetrating through the brain. And the blood-brain barrier is obviously on all of those blood vessels, because it relates to this exchange between the blood itself and the CSF. So the blood-brain barrier is on the surface of all of those blood vessels that are penetrating through the brain. And the difference between the endothelial cells that are lining the blood vessels within the brain versus the endothelial cells that are lining the blood vessels everywhere else is that they have much tighter junctions between them. And so they have very selective tight junctions that are in the gaps between these endothelial cells. And so that basically means that only very tiny molecules can penetrate through these tight junctions between the endothelial cells. Everything else has to pass through other cells. And we also have a thick basement membrane, and we also have projections from astrocytes that act to come down to these endothelial cells and kind of reinforce this barrier that's acting to separate the nervous system from the blood. And anything that's sort of large is obviously going to have to pass through the endothelial cells and then through some of these other cells. And because they have to go through cells to get to the CSF, the cells actually have the ability to regulate what gets in. And the way it does that is through a lot of transport processes. So for example, there are a lot of situations where you have, you can get a lipophilic compound, and it has to be lipophilic so it can diffuse through membranes. And it can get the first step of getting out of the blood into these the endothelial cells and the basement membrane and so on. But they're full of transporters, which in many cases are just pumping things back into the blood. So they can diffuse into these cells and then get pumped back into the blood. And so in that way, the blood-brain barrier does an amazing job of keeping things out of the brain. This is also significant because, like if you actually want to get something into the brain, that means you have significant challenges to get it in there. And Dr. Yang and Dr. Bai will sort of talk about some of that in their classes. So this is a cartoon of what's going on in the blood-brain barrier. So we have our endothelial cells of the blood vessels, pretty much like any other, like the outside of any other blood vessel, except that they have particularly tight junctions. But then we also have this basement membrane, and then we have the astrocytes acting sort of to reinforce that, and then these other specialized cells, pericytes, which also act to reinforce the sort of structure of the blood-brain barrier and keep the blood separate from the CSF. If you have something like multiple sclerosis, what basically is happening is that in some way there has been damage to the blood-brain barrier, and that's allowing immune cells from outside the nervous system to get in, and that's what causes the damage to the neurons that leads to all of those symptoms. So that's a little bit of the sort of outside of the nervous system, but the nervous system can be broken down into many different ways. And so this is just a kind of hierarchical sort of chart of how how you can sort of separate the bits of the nervous system. So we have the central nervous system, which is the brain and the spinal cord. And then we have the peripheral nervous system, which is everything that's nervous that is not part of the central nervous system. So it's basically everything that comes out of everything that's outside of the meninges is going to be the peripheral nervous system. And obviously there has to be some way for information to get out of the central nervous system, otherwise obviously it would be pretty useless because it would just sit there and might think great thoughts, but it wouldn't be able to sort of do anything to sort of, interact with the world and the body. So the peripheral nervous system is basically where everything, all of the outflow of the central nervous system, but also all of the sensory information that's got to come into the central nervous system in order for it to do anything. So we have our sensory system, which is the sort of and these are called afferents because they're sort of information is coming in that way. And when we have our motor system, and these are often called efferents, so that's now information's coming out from the nervous system. It's these are projections out from the nervous system. And then the motor system can then be a subset of that is going to be the somatic nervous system, which is sort of basically just voluntary movement. This is how we're able to move our muscles and so on. The autonomic nervous system is sometimes also regarded as a branch of the motor system. And the autonomic nervous system is responsible for the regulation of basically all of physiology. So everything, whether it's your digestion, your heart rate, your breathing, all of those things are regulated by the autonomic nervous system. And so there are subsets of the autonomic nervous system. We have our sympathetic nervous system, which is responsible for the sort of fight or flight type responses. And we have our parasympathetic nervous system, which is sort of more, well, is the one that people say rest and digest. So it's the kind of opposite to fight or flight. And then we also have our enteric nervous system, which is its own semi-autonomous nervous system responsible for sort of regulating everything that goes on with the stomach. It's the whole GI tract. And I talk very, very little about it. Although I think I am taking over next year, I will have taken over the lectures on antiemetics and stuff like that, because it sort of kind of fits more neuro than it does. So that last slide is the sort of hierarchical breakdown of like terminology essentially for the nervous system. If we're looking more anatomically, one of the major things we have to consider are cranial nerves. So the cranial nerves are essentially just nerves that are coming out from the CNS at a level above the spinal cord. And in many cases, these are sort of involved in the parasympathetic nervous system. And I have highlighted 3 of the cranial nerves. Like all of them are sort of kind of important, but these are the 3 that are highlighted sort of like most likely that I would ask questions on, because I feel it would be a little unfair to ask about all 12 of them, especially things like motor control of the tongue. It's like probably not going to come up very often in your clinical careers. So, but if you're trying to sort of assess neurological damage, they all become significant. But so the vagus nerve is the sort of like the star of the cranial nerves, because the vagus nerve is the nerve that is responsible for the parasympathetic input to the like the heart and the lungs and so on. The optic nerve is obviously the one responsible for bringing vision to the brain, and the vestibulocochlear nerve is responsible for hearing and balance. And this picture is just to sort of give you an illustration of how it is organized sort of anatomically, although like absolutely no question you will not have a sort of like label this diagram type question from me. So the next bit of anatomy we need to worry about is the spinal cord. And so the spinal cord has a segmental organization, which, obviously, as you would imagine, probably imagine that just from, looking at a spine where you have all of those little individual vertebrae. And they have the sort of inputs and outputs, the afferents and efferents, grouped sort of, like loosely together. And each segment of the spinal cord is defined by a posterior root entering it and an anterior root exiting it. And so these nerve roots are just basically bundles of nerves so that they're big enough that you can see them by eye. Because obviously, when they get down to sort of individual axons, you obviously can't see them by eye because they're sort of like a micron across. If you do a cross-section through the spinal cord, you'll see lighter and darker areas. And so the lighter areas are white matter that is predominantly myelinated axons going up and down the spinal cord. And the darker areas are where the neuronal cell bodies of the spinal cord are located. So you have this sort of dorsal horn and the ventral horn. And the dorsal horn is going to be the cell bodies of different types of sensory neurons. So they're receiving information from outside of the spinal cord, and then they'll be sending axons up, like to the brain to sort of say, oh, I felt something in my little finger, something like that. The ventral horn is where the cell bodies of the motor neurons are going to be seated. So that's and that's predominantly the sort of actual somatic motor neurons, the ones responsible for sort of muscle contractions. And then this region of in the middle between them, which is the lateral gray column, is going to be where most of the sympathetic nerves, cell bodies, are going to be located. So, these are sort of obviously, going to be sending outputs saying sort of, make my heart beat faster because I have to run away from something scary. So that's basically that overall pattern is going to be repeated all the way up the spinal cord, but there are specific, depending on your level of the spinal cord, will obviously reflect like what particular things are going to be impacted. So and obviously if you sort of get low down in the spinal cord, there's sort of almost nothing there. It's like super thin, and that's why, for example, you do lumbar punctures way down in the spinal cord because you're less likely to damage anything. And this is illustrating that point where like the inputs and outputs change as you descend through the spinal cord. And we will come back to this. This is not something you need to worry about for today's class, but this is the organization of the autonomic nervous system, the inputs and outputs there, where you can see for the parasympathetic inputs and outputs, they're like through these cranial nerves way up top. The sympathetic outputs descend much further through the spinal cord. And then we'll just spend the last couple of minutes giving an overview of the anatomy of the actual brain. So we have it's usually broken into sort of, say, 4 major regions. So we have the hindbrain, the midbrain, the forebrain, and then the cerebral hemispheres. So the hindbrain is contains the pons, the medulla, and the cerebellum. And this is where a lot of the housekeeping stuff occurs within the brain. So this is going to be important for sort of keeping us breathing, keeping our heartbeat going, having some of the reflexes that regulate this stuff. The cerebellum is important in the coordination and integration of movement. And overall, the hindbrain is sort of the evolutionarily most ancient part of the brain. So this is where people are sort of like, when people are talking about their lizard brains and things like that, this is sort of kind of where some of that stuff is coming from. The midbrain is sort of like a little bit further up from the spinal cord. And this is where we have things like our substantia nigra, which is involved in regulation of movement. We have a ventral tegmental area, which is involved in reward, for example, and raphe nuclei, which are involved in mood. Then we have the forebrain, and this has some very important areas. So we have the thalamus, the hypothalamus, the hippocampus, and the amygdala. And so the thalamus is the input-output station of the brain. It's going to be regulating the ability. So all of the sensory information that actually gets through to the brain will go through the thalamus. The hypothalamus is completely different, even though it has a similar name. And the hypothalamus is generally involved in regulation of physiology. So it's regulation of all of the autonomic functions of the brain. It's sort of keeping, like the body going. The hippocampus is one of the most central areas of the brain involved in memory. And then the amygdala is primarily responsible for the processing of emotions. And then we move on to the cerebral hemispheres, which are sort of basically on the outside of all of that other stuff. And they are broken down into these different areas. And so we have our frontal lobe, which contains both the olfactory bulb, and obviously involved in sense of smell, but also the motor cortex, which is a little bit further back. involved in sort of obviously direction of movement. Then we have the parietal lobe, which is behind that. So we have frontal lobe, parietal lobe. The parietal lobe, probably most important for housing the sensory cortex, which is obviously where we have our projection of all of our sensations that come in. Then we have the temporal lobe, which is kind of lower down, like here. And that's involved especially in the processing of sound and speech. And then we have the occipital lobe, which is right at the back, which is where we have vision. And this is why if you fall over and smack your head on the back, you get a flash of light. It's because you've just banged your occipital lobe. So that's where the visual cortices are found. And we don't really have time for the 2-minute thing. So you can try and maybe do that in your head, or feel free to sort of discuss it while you're waiting for, is it biochemistry next?
Yep.
So, and then, as I said, these study questions are aimed to sort of try and help you sort of like view the sort of class materials through these questions. And so to sort of, so we're kind of all on the same page as to sort of what I think I've been talking about for the last, like 50 minutes. And